Hello and welcome back to the Biochemistry for Health Sciences channel. Today's video is starvation, amino acids for gluconeogenesis. As mentioned in previous videos, gluconeogenesis is a very important metabolic pathway, especially during starvation. Gluconeogenesis requires energy, and most of this energy comes from fatty acids. Energy is not a problem because even during starvation, we have sufficient fat that can give us fatty acids that can provide energy for gluconeogenesis. However, gluconeogenesis also requires carbon skeletons, building blocks to make new glucose. And these carbon skeletons include lactate, which can be converted to pyruvate or pyruvate to lactate through the enzyme lactate dehydrogenase. Another important precursor or carbon building block of gluconeogenesis is glycerol, and the third one is amino acids. In this video, we shall focus on amino acids as carbon building blocks for gluconeogenesis. Before we go further, I should mention that this is a complex topic, carbon skeletons of amino acids for gluconeogenesis, because for many reasons. One, it involves many amino acids. Number two, when we say the carbon skeletons of amino acids, do these carbon skeletons really come from amino acids? Because these carbons can exchange with non-amino acids such as lactate, glycerol, as well as glucose. In addition, this topic involves many organs, especially the skeletal muscles, as well as the liver, kidneys, and intestine. There are several steps that are still uncertain in scientific literature, and for that reason, we cannot tell you a story on this topic as we have done in many of our previous videos. So what we shall do is we will focus on the major concepts. So the first important question is, where does gluconeogenesis occur in our body? And as far as what we know today, the major organs where this metabolic pathway happens is the liver, which is a predominant organ during the early part of starvation. And then we have the small intestine, which has a lesser contribution to gluconeogenesis, also during the early part of starvation. And finally, we have the kidneys, which is the predominant organ for gluconeogenesis, especially during the later part of starvation, or what we refer to as prolonged starvation. So we are going to talk about liver, small intestine, as well as the kidneys as the major sites for gluconeogenesis. In the future, we might find newer sites. However, for this video, these are the three sites that we'll talk about as far as gluconeogenesis is concerned. So where do these amino acids that provide the carbon skeletons for gluconeogenesis come from? And the answer is anywhere in the body, including the gluconeogenesis organs that make glucose from these carbon skeletons. However, since most of our proteins, which means most of our amino acids, are in skeletal muscles therefore are a significant contributor to the gluconeogenesis precursors in the form of amino acids. Any amino acid that can be broken down into either pyruvate or oxaloacetate or any intermediate such as alpha-ketoglutarate or succinyl-CoA, which can be converted into oxaloacetate. So any amino acid that will give us pyruvate, oxaloacetate, or something that be converted to oxaloacetate can be used for gluconeogenesis. These amino acids then can give us our carbon skeletons for gluconeogenesis. Out of 20 amino acids that we find in proteins, 18 amino acids can provide carbon skeletons for gluconeogenesis. Two amino acids, leucine and lysine, when we break them down, we get either acetoacetate or acetyl-CoA. Humans cannot convert acetyl-CoA or acetoacetate into pyruvate or into oxaloacetate, and therefore these two amino acids are what we call purely ketogenic amino acids, lysine K and leucine L. So these are the two amino acids that cannot provide our body with carbon skeletons to make glucose. A mixed amino acid is an amino acid that can give you either acetoacetate or acetyl-CoA, which cannot be made into glucose, but it can also give you something that can be made into glucose. So for example, isoleucine. You can see isoleucine gives you acetyl-CoA, which cannot be made into glucose. However, isoleucine also gives you succinyl-CoA, which can be converted to oxaloacetate and then be used to make glucose. So isoleucine is an example of a mixed amino acid. And then we have what we call purely glucogenic amino acids. So for example, valine. You can see valine, when we break down valine, we only get succinyl-CoA. We do not get acetyl-CoA or acetoacetate. So amino acids such as valine, these are called purely glucogenic amino acids. So an important message here is that we can use most of our amino acids, 18 out of 20, to make glucose by gluconeogenesis. Since the bulk of proteins are in our muscles, and when we break down proteins, we get amino acids. That happens during starvation. And in one of previous videos, we talked about how proteins are broken down into amino acids by the ubiquitin proteasome system as well as autophagy. So by these processes, especially during starvation, muscle proteins are broken down into amino acids. Now, how are these amino acids transported to the gluconeogenesis organs? They are mostly transported as alanine and glutamine, as well as a few others. So let's first see how does the skeletal muscle make alanine. So during starvation period, muscle proteins are broken down into amino acids. Then in the mitochondria, the amino acid undergoes a transamination reaction catalyzed by enzymes called transaminases. Basically, the amino acid will react with an alpha keto acid. The amino acid will become an alpha keto acid, while the alpha keto acid will become an amino acid. So if the alpha keto acid is alpha ketoglutarate, then the amino acid that is made would be glutamate. So basically here during this transamination reaction, the amino group from the amino acid is being transferred to the alpha ketoglutarate, and the alpha ketoglutarate becomes glutamate. So now this glutamate will carry the amino group from this amino acid. The glutamate will now go through another transamination reaction that is catalyzed by the enzyme AST, which stands for aspartate transaminase. The old name for this enzyme was glutamate oxaloacetate transaminase, or GOT. In this reaction, the amino group from the glutamate is transferred now to the alpha keto acid oxaloacetate, and the oxaloacetate will now become aspartate. So you can see the amino group joined to the oxaloacetate to form the new amino acid aspartate. Glutamate, after it loses its amino group, will become alpha ketoglutarate. The alpha ketoglutarate as well as the aspartate will be transported out of the mitochondria into the cytoplasm. In the cytoplasm, the cytoplasmic form of the enzyme AST will now do the reverse reaction, basically take alpha-ketoglutarate and aspartate and make it into oxaloacetate and glutamate. And that's what we see here. Glutamate and oxaloacetate are reformed in the cytoplasm. So now this glutamate that is carrying the amino nitrogen, this glutamate now undergoes another transamination reaction that is catalyzed by an enzyme ALT, and that stands for alanine transaminase, or the old name for this was GPT, which stands for glutamate pyruvate transaminase, ALT or GPT. In this reaction, the amino group of glutamate is transferred to pyruvate. The pyruvate becomes alanine, and the glutamate becomes the alpha-keto acid, alpha-ketoglutarate. Now it's important to note that this pyruvate here does not come from amino acids, or most of this pyruvate carbon skeleton does not come from amino acids. Rather, it comes from the breakdown of muscle glycogen into glucose 6-phosphate, and that glucose 6-phosphate will go through glycolysis to give you pyruvate. So this alanine that is formed will then be sent out into the blood, and most of it will be used by the liver for gluconeogenesis. So it's important to note once again that the carbon skeleton that we see in alanine that is coming from the skeletal muscles, this carbon skeleton is not coming from an amino acid. It's really coming from pyruvate, which is mostly coming from muscle glycogen. So that's how the skeletal muscles will use the nitrogens from amino acid breakdown as well as the pyruvate from glycogen to make quite a bit of alanine, which is then sent to the gluconeogenesis organs, and most of the alanine, as we shall see later, will be used by the liver to make new glucose via gluconeogenesis. Now let's see how the skeletal muscle makes the amino acid glutamine. Just as in the case of alanine, the amino acids that are derived from the breakdown of muscle proteins undergo a transamination. The amino group is transferred to glutamate, and the glutamate then undergoes the transamination by AST. Glutamate and oxaloacetate will react to give you alpha-ketoglutarate and aspartate. These two are transported out of the mitochondria into the cytoplasm, and once again they are reconverted back into glutamate and oxaloacetate by the cytoplasmic form of aspartate transaminase. Now, unlike alanine, the glutamate doesn't go through the ALT reaction. Instead, it is converted into glutamine by the enzyme glutamine synthetase. So let's look at glutamine carefully and see where the different parts of glutamine come from. Okay, so we have this amine group that's connected to the alpha carbon. So this is your alpha amino group. And then we have this amine group that is connected to the R group of glutamine. This is in the form of an amide. And then we, of course, have the carbon skeleton. Okay, we have the carbon skeleton of glutamine. So let's see where these different groups come from in case of glutamine that is made in these skeletal muscles. So the alpha amine group, this group, if we trace this group, it really comes from any amino acid. So you can see the alpha amino acid, the alpha amine group of any amino acid is transferred to glutamate. So here is the alpha amine group, and then that's transferred to aspartate, and then the reverse of the reaction happens in the cytoplasm, and once again we get glutamate. So this is the amine group of glutamate, which is this group here. Okay, so basically this group is the alpha amine group from the amino acids that are broken down in skeletal muscles. Next, let's look at this amine group that is part of the R group of glutamine. Where does this amine group come from? Now, skeletal muscles have this enzyme called myokinase, and this enzyme can basically convert ADP into ATP. And one of the products that is generated is AMP. The AMP then enters what we call the purine nucleotide cycle. And in the cycle, AMP is broken down by the enzyme AMP deaminase into IMP and ammonia. The IMP will react with aspartic acid. So aspartic acid and IMP will react to form adenosuccinate. And this adenosuccinate will then break down to AMP, giving you fumarate. So this part here is called the purine nucleotide cycle that happens in the skeletal muscles. The enzyme glutamine synthetase will react this ammonia together with glutamate, and that gives you this amine group, which is part of the amide that is in glutamine. Okay, so we have this amine coming from the amino acid, any of the amino acids that are being broken down, and then we have this amine that's part of the R group, part of the amide, coming from ammonia. Now let's look at the carbon skeleton. Okay, so let's look where this carbon skeleton is coming from. Recall that the carbon skeleton in case of alanine actually came from pyruvate, and that pyruvate came from glycogen. Okay, not an amino acid, but glycogen. So in case of alanine, the carbon skeletons are mostly from glycogen. However, in case of glutamine, there are several amino acids such as aspartic acid, asparagine, glutamic acid, valine, and isoleucine that are known to actually give a net generation of carbon skeletons such as alpha-ketoglutarate and oxaloacetate. So the oxaloacetate and the alpha-ketoglutarate actually comes from the amino acids. So this is where the carbon skeletons come from. So alpha-ketoglutarate, which is part of glutamate, will give you the carbon skeleton for glutamine. Now glutamine with its alpha amino group from the amino acids, the carbon skeleton also from the amino acids, and this amine of the amide from ammonia, this glutamine will now be transported out of the skeletal muscles towards the gluconeogenesis organs, and as we shall see later, most of the glutamine is used by the kidneys as well as the small intestine. A few words about the branched chain amino acids, BCAAs. That includes valine, leucine, as well as isoleucine. Now, branched chain amino acids form almost 20% of your proteins in your diet. and about 15% of muscle protein amino acid is actually branched chain amino acids. The metabolism of BCAAs, branched chain amino acids, is shown here. Basically, there's a transamination reaction using branched chain amino transferases or BCATs that will give you branched chain alpha keto acids. These are the branched chains alpha keto acids. And then the enzyme BCKD or branched chain alpha keto acid dehydrogenase will basically oxidize these branched chains into acyl-CoA. So coenzyme A is required. So we get the different acyl-CoAs. So isoleucine will give this keto acid and this acyl-CoA. Valine will give this keto acid and this acyl-CoA, while leucine will give this keto acid and this acyl-CoA. Then the acyl-CoAs undergo further metabolism and give you acetyl-CoA as well as acetoacetate or succinyl-CoA. So in case of isoleucine, we will get acetyl-CoA that makes isoleucine a ketogenic amino acid, but we also get succinyl-CoA, which can be converted to glucose. So since isoleucine can give you acyl-CoA as well as something that can be made into glucose, in this case succinyl-CoA, isoleucine is a mixed amino acid. Valine gives you only succinyl-CoA. Therefore, valine is your purely glucogenic amino acid. Leucine will give you only acetyl-CoA and acetoacetate. And both of these cannot be converted into glucose. So leucine, as we mentioned earlier, is a purely ketogenic amino acid. So that's basically how these branched chain amino acids are metabolized. Transamination to give you keto acids, and then the dehydrogenase, the branched chain alpha keto acid dehydrogenase to give you acyl-CoAs, and then the respective acyl-CoAs are broken down into the products, succinyl-CoA, acetyl-CoA, as well as acetoacetate. When muscle proteins are broken down into branched chain amino acids, there are several things that can happen to these branched chain amino acids that are formed from breakdown of muscle proteins. One is that the branched chain amino acid may be transported out of the muscle cell to the rest of the tissues. Since muscle cells are very rich in BCAT enzyme, these branched chain amino acids could be converted into their keto acids. And then these keto acids could also be exported out of the muscle cells into the bloodstream, and then from there to all the different tissues, including the gluconeogenesis organs. A third possibility is the branched chain amino acids are simply oxidized into succinyl-CoA, acetyl-CoA, and acetoacetate, and all of these which could be used by the skeletal muscles. Another possibility is that the branched chain amino acid can be made into glutamine. So this glutamine, as we mentioned earlier, for glutamine synthesis in muscles, we'll have the amine group, the alpha amine group from the branched chain amino acids. The ammonia will give you the amide group of glutamine, and the carbon skeletons will come from the branched chain. So this glutamine will be exported out of the skeletal muscles to the gluconeogenesis organs, especially the kidneys and the small intestine tend to use more glutamine than alanine. And the fifth possibility is that the branched chain amino acids will give the alpha amino group for alanine, while the pyruvate will come from glycogen, and That alanine will be exported out of the skeletal muscles and especially will be used predominantly by the liver, small extent by the kidneys and the intestine. Okay, so this is how the skeletal muscles make the amino acids that come from the breakdown of proteins in the skeletal muscles, either into alanine or glutamine, or it could transport some of these amino acids to the gluconeogenesis organs. Now let's see how do these gluconeogenesis organs actually use these amino acids, especially alanine and glutamine. Let's first look at how alanine from tissues such as skeletal muscles are transported to the liver. That's where most of the alanine is used, and the liver will then take the carbon skeleton and use that to make glucose by gluconeogenesis. And then that nitrogen, that excess nitrogen will be converted into urea, and that urea will be sent to the kidneys. If there's excess nitrogen, the kidneys will excrete the urea into the urine. So the alanine that comes from the skeletal muscles as well as other tissues, but predominantly from the skeletal muscles during starvation, will enter the liver cells, the hepatocytes, and there the alanine will react with alpha-ketoglutarate to form pyruvate and glutamic acid. And this is a transamination reaction catalyzed by alanine transaminase. The pyruvate from the cytoplasm will be sent to the mitochondria, and the same thing happens to glutamate. So now we have glutamate and pyruvate in the mitochondria of the hepatocyte. The pyruvate is next carboxylated by the enzyme pyruvate carboxylase, and this pyruvate will now become oxaloacid. Basically, we're adding a carbon dioxide to the pyruvate, to this part here, and that gives us oxaloacetate. So now we have the pyruvate carbons that came really from the skeletal muscles. So we have the pyruvate carbons now in the form of oxaloacetate. In the next reaction, the oxaloacetate will react with glutamate, and that gives you alpha-ketoglutarate and aspartate. And this reaction again is a transamination reaction, in this case catalyzed by aspartate transaminase. So now we have the carbon skeletons in aspartic acid. This is exported out of the mitochondria, the aspartic acid, and this goes through a transamination reaction to give you oxaloacetate. So basically the same AST enzyme, but this case the cytoplasmic form will react aspartic acid with alpha-ketoglutarate, giving you glutamic acid and oxaloacetate. This oxaloacetate then can be used to synthesize glucose by gluconeogenesis. And this glucose then will be transported out into the bloodstream in order to maintain the blood concentration of glucose at optimum levels, more than at least 40-50 milligrams per deciliter during starvation. What about the nitrogen part of the alanine? You can see the nitrogen in this transamination reaction has now become part of glutamic acid. So that's the glutamic acid. And now this glutamic acid is converted to alpha-ketoglutarate by the enzyme glutamate dehydrogenase 1, which is the one that's more predominant in liver cells. And what happens is this nitrogen comes out as ammonia. So basically now we have the nitrogen in the form of ammonia, and the liver will now convert this nitrogen or incorporate this nitrogen into the organic compound called urea. So basically the ammonia will react with the bicarbonate to give you carbonyl phosphate, and this then enters the urea cycle. This is the urea cycle, and out of the urea cycle comes urea, and urea has this nitrogen that really came from alanine. So in this way, basically the carbon skeleton of alanine is made into glucose, while the amino group of alanine will be made into urea, and this urea will be sent to the kidneys, and there it can be excreted into the urine if there is excess nitrogen in the body. Now let's see how the small intestine uses amino acids, especially glutamine, to make glucose via gluconeogenesis. But besides glucose, it also generates alanine as well as lactate, and the nitrogen comes out as ammonium. So how does this happen? Glutamine is transported into the enterocyte, that's the cell of the small intestine that carries out gluconeogenesis. So the glutamine first undergoes a reaction that is catalyzed by glutaminase. So basically this amine group that is attached to the amide comes out as ammonia, and the glutamine gets converted to glutamic acid or glutamate. The glutamate then goes through a transamination reaction catalyzed by AST, basically reacts with oxaloacetate, giving you alpha-ketoglutarate and aspartate. This aspartate is then transported out of the mitochondria, and then it undergoes the same GOT or AST reaction, giving you back oxaloacetate. This oxaloacetate is then converted to glucose, which is then sent out into the bloodstream. So this is where the gluconeogenesis happens, and the small intestine can also contribute to glucose levels in the blood. Notice that on the way to making glucose, the phosphoenol pyruvate can also be converted into pyruvate by the enzyme pyruvate kinase. And this pyruvate can be made into lactate, and the intestine is also known to secrete considerable amounts of lactate, which is also, as you remember, is a gluconeogenesis substrate. So this lactate can go to gluconeogenesis organs, and especially the liver, and be used to make glucose if necessary. The pyruvate can also go through transamination reaction in the enterocytes, and that gives you alanine, which is exported out into the bloodstream. So the kidneys the small intestines do produce glucose, lactate, as well as alanine. The small intestine also has low activity of glutamate dehydrogenase, the isoform 2, the GDH2. So some of the glutamate could be converted to alpha-ketoglutarate, releasing ammonia. And then this alpha-ketoglutarate is converted to malate, which is sent out of the mitochondria, and that malate is converted to oxaloacetate by malate dehydrogenase, and this oxaloacetate can then be made into either glucose or pyruvate or lactate or alanine. So you can see that the carbon skeleton of glutamine will appear as glutamate after this reaction, and after the transamination reaction we'll have alpha-ketoglutarate, which by the TCA cycle can be converted to oxaloacetate, and then this oxaloacetate carbons now basically appear in aspartate, and that aspartate carbons will regenerate oxaloacetate, which will then come out into glucose. The nitrogen that is attached to the glutamine via the amide in the R group, that comes out as ammonia in this reaction, the glutaminase reaction, and this ammonia is then secreted out into the bloodstream. The amino, the alpha amino group of glutamine here, you can see appears as glutamate, and in this reaction, glutamate dehydrogenase can be liberated as ammonia, which can then be secreted out of the enterocyte, and this ammonia from the small intestine will be transported to the liver, and in the liver the ammonia is converted into urea, which can then be excreted out into the urine by the kidneys. Now let's see how the kidneys use gluconeogenesis precursors, especially glutamine. Kidney uses a lot of glutamine, uses only about 10% of alanine, since the kidney is very poor in the enzyme alanine transaminase. So kidneys, the cortex part of the kidneys, not the medulla, okay, the cortex part of the kidneys, they do carry out quite a bit of gluconeogenesis. In fact, might be one of the major organs for gluconeogenesis, and it uses predominantly glutamine. So that glutamine from the skeletal muscles and elsewhere will enter the kidneys. We'll see that the carbon skeleton will be used to make glucose, while the two amines, the alpha amino group as well as the amine attached to the R group, they will be basically will come out as ammonia. But now the kidneys, they can excrete this ammonia, okay, together with acid, with H+. We'll see that this ammonia and H+ can come out into the urine as ammonium. It doesn't have to be converted to urea, because it's safe to get rid of this ammonia out as urine. It's not going into the bloodstream like urea is. Okay, so besides producing or getting rid of excess nitrogen in the form of ammonium, making glucose to maintain our blood glucose concentration during starvation, we'll see that the kidneys also produce bicarbonate, and this bicarbonate is very important to buffer the fatty acids and the ketone bodies that are predominant during starvation. Okay, so the kidneys play a very, very important role in starvation, not just providing glucose to maintain our blood glucose, but getting rid of excess nitrogen because of all the proteins that are being broken down, and also helping buffer the fatty acids and the ketone bodies, both of which are acids that are accumulating or that are increasing as one gets into prolonged starvation. So the reactions in the kidneys is very similar to what we see in the small intestine. Glutamine enters the kidney cells in the cortex. So we have glutamine, and then it undergoes glutaminase reaction. This amine of the amide is released as ammonia. We get glutamate. This glutamate undergoes the GOT transamination reaction, gives you aspartate. Aspartate comes out into the cytoplasm, reconverted into oxaloacetate, which is then made into glucose via gluconeogenesis, and that glucose is sent out into the bloodstream to maintain our blood glucose concentration. So while this nitrogen comes out in glutaminase reaction, the alpha amino nitrogen comes out when glutamate is converted to alpha ketoglutarate by glutamate dehydrogenase 2, which is very active in the kidneys. So this nitrogen basically appears as ammonia in this step. So this ammonia as well as this ammonia now will be excreted out into the urine. Okay, so we see the ammonia being excreted out into the urine. Through this carbon dioxide reacting with water to this reaction to form carbonic acid, that's broken down into acid, protons, and bicarbonate. Through this reaction, the kidneys can also excrete or secrete acid. And this acid will combine with ammonia to form ammonium, which can be excreted out into the urine as ammonium salts. So in this way, the kidney can get rid of excess nitrogen directly into the urine. Now, the bicarbonate that is formed in this reaction will be secreted into the bloodstream. And there, the bicarbonate, as we shall discuss in a future video under acid-base balance, bicarbonate will help buffer all the fatty acids that are being produced during late starvation, as well as the ketone bodies, which are also acids. And so this bicarbonate will be very important in maintaining the blood pH at 7.4 in order to make sure that the blood pH doesn't drop because of all these acids that are being produced during starvation. So the kidneys play a very, very important role in starvation. You can see that not only does it give us the buffering capacity, it gets rid of excess nitrogen, and then it produces glucose via gluconeogenesis, so that our blood glucose concentration is maintained more than 50 milligrams per deciliter, so that there is enough glucose for the brain during prolonged starvation. So as starvation continues, we can see that the liver gluconeogenesis decreases. Okay, so from 85% of blood glucose that is supplied by liver, it goes down to 55% as starvation progresses, while kidney gluconeogenesis starts increasing. So from 15% of blood glucose that is supplied by the kidneys at the beginning of starvation, in later parts of starvation, almost as much as half of the blood glucose is supplied by the kidneys. Now, since the liver excretes excess nitrogen as urea, while kidneys excrete excess nitrogen as ammonium, we can see that the concentration, the amount of urea in the urine also keeps on decreasing during starvation. So at the beginning, there's a lot of urea in the urine, but in later parts of starvation, we see less urea and more ammonium in the urine. So when the concentration of ketone bodies get very high, remember ketone bodies are small, they are very water soluble. So ketone body like beta-hydroxybutyrate, when it's present in very high concentrations, it basically saturates the proteins that can reabsorb the beta-hydroxybutyrate. This protein is SMCT, sodium coupled monocarboxylate transporter. So at the beginning, we do see some quite a bit of ketone bodies being lost into the urine. So for example, remember the urine has ammonium salts as coming from the kidneys, and now we have hydroxybutyrate or acetoacetate. So ammonium hydroxybutyrate or ammonium acetoacetate would be excreted out at the beginning of starvation. But since ketone bodies are very important, because now, remember, in prolonged starvation, it's providing energy to the brain. So gradually, over time, the kidneys will start making more of this transport proteins, and that will increase the reabsorption of the ketone bodies back into the bloodstream. So that way, the body will decrease the loss of these very essential ketone bodies in later parts of starvation. And eventually, as starvation progresses, we reach a sort of a steady state where the beta-hydroxybutyrate concentration is about 10 millimolar in the bloodstream. Glucose concentration is usually in the normal to low range. So we're talking about around 50, 60 milligrams per deciliter, while the bicarbonate concentration is on the low side, about 18 millimolar, instead of the normal of about 24 millimolar. So to summarize, we can see that starvation, the key thing is we want to make sure that the brain has sufficient energy. So at the beginning it will use glucose, and then later as starvation progresses, it will start using more and more ketone bodies. Now the red blood cells can only use glucose. They cannot use ketone bodies, so they will continuously use glucose. Where does this glucose come from? It comes from the gluconeogenesis organs. That includes your liver as well as the kidneys. Those are the two most important, and a small contribution by the small intestine, the enterocytes. And another thing we see in the later parts of starvation is the accumulation of ketone bodies. They will keep on rising because these ketone bodies are very important for the brain to be used as fuel. One of the problems with accumulating ketone bodies is ketoacidosis. Although these ketone bodies are weak acids, when they are produced in very large amounts and our blood doesn't have enough buffering capacity, that can actually decrease the blood pH. Okay, and this can be very, very dangerous because, as we shall discuss in future videos, on acid-base balance, a sudden decrease in blood pH can lead to serious consequences, even death. And recall, while the red blood cells at this stage are using only glucose, the brain is using glucose and ketone bodies. The rest of the tissues, including the liver, will be using fatty acids, and the kidneys will be using fatty acids, and the adipocytes will be using fatty acids, and especially the muscles, the skeletal muscles, the heart muscles, they all will be using fatty acids as well as ketone bodies. Okay, so everyone can use ketone bodies except the red blood cell. So this is how starvation occurs, the different stages of starvation, a very complex process where we are still trying to learn a lot about. Hopefully in the next few years we'll understand more about this very important process that has a lot of relationship or association with the diseases that we see today, including diabetes as well as obesity. Thank you for watching, and until the next video, have a wonderful day, and hopefully we'll see you soon.